Good morning. I am Mark Roithmeier. I have the privilege of being the CEO of the Alzheimer's Drug Discovery Foundation. We are delighted that so many of you showed up this morning. Thank you, because this is our third annual Palm Beach Luncheon, but it is the first time we're doing a symposium to start the luncheon because we feel we have so much exciting and relevant information to share with you all about where we are in prevention and drug discovery and potential new therapies to treat Alzheimer's. So just as a quick reference, 22 years ago, Leonard and Ronald Lauder started this fabulous organization, the Alzheimer's Drug Discovery Foundation. In the last several years, there's been a lot of excitement. I think those of you, how many folks were here last year or the year before? There we go. Nice to have you back. And for all the new folks, it's fabulous to have you with us today. We're very excited at the foundation for a lot of reasons, and you'll hear that from Dr. Howard Phillip and Dr. Ron Crystal today. But certainly wind in our sails in the last couple of years has been Bill Gates and Jeff and Mackenzie Bezos have come to us. They've invested in the ADDF. We have an exciting program called the Diagnostic Accelerator that's looking to find a blood test so that we could test through a simple blood test for Alzheimer's and dementia in people. So you'll hear much more about that. We have a second round of funding on that program coming out, and it's going to be for digital biomarkers. So how can we use technology, right? And this is coming from folks, Bill Gates and Jeff and Mackenzie Bezos, so we have an excellent partnership there. Like I said, you'll be hearing a lot more about all of this from Drs. Phillip and Crystal. I have the privilege right now of bringing on Dr. Howard Phillip. He is the Chief Science Officer and the founding Executive Director of the ADDF. He was there at the beginning with Leonard and Ronald and helping to point out where this ship was going to go, and here we are 22 years later. He is a genius on the science, but he also has a true north, and that true north are the patients that he still continues to see, and how can we improve the lives of those who have cognitive issues. So ladies and gentlemen, to start off our symposium today, I give you Dr. Howard Phillip.
Thanks, Mark. Thanks, Mark. And I want to thank all of you for coming today to be with us, and I hope that you'll ask a lot of questions. Feel free to ask any question that you have during the symposium once we make our presentation. As Mark mentioned, I'm a geriatrician by clinical training. I've been taking care of people with Alzheimer's disease for over 40 years. I know the disease pretty well. My own dad died of it not too long ago. And I'm also a neuroscientist, and I've been in the research on Alzheimer's from the very beginning, back in 1979 or so when we started. Back then, we knew basically nothing about Alzheimer's, and there's a lot of historical reasons for that. Cancer research started around 1900. Alzheimer's research really didn't start till about 1980. So even though we've had a lot of failures, and that's the nature of developing drugs, 22 years later, after having started the ADDF, I've really never been more excited about where we are today in the last 40 years. The tremendous strides that we've made going from literally knowing nothing about the disease to knowing what we know today. There's over 100 drugs in clinical trials. I'm proud to say that the Alzheimer's Drug Discovery Foundation we have our fingerprint on about 20% of those drugs. We currently have a very strong focus on human research in Alzheimer's and particularly clinical trials. As Mark mentioned also, we have better diagnostics today. In the old days, it was basically impossible to make a definitive diagnosis of Alzheimer's disease. The only way you could really do it was at autopsy, and most of my patients didn't want to go to autopsy, so we just didn't do that. Today, there's a brain scan. I can just write a prescription. If somebody comes into my office with some memory problems and they really want to know what's going on, I can write a simple prescription for a PET scan, a brain scan. They can go down the block to the radiology office, and by the next day, we can know with about 98% certainty whether the patient has Alzheimer's disease. The way the brain scan works is it detects the plaques that are characteristic of Alzheimer's disease, what Alzheimer described back in 1906. Now that brain scan really has been termed by the neurological community as a revolutionary technology. It changed early diagnosis. Now we can make a definitive diagnosis in the clinic. It changed the way we do clinical trials, and it changed the world of prevention studies for Alzheimer's disease. And I'm proud to tell you that that brain scan, which was the first diagnostic test approved by the FDA for Alzheimer's disease, was enabled and funded and created in part by our Alzheimer's Drug Discovery Foundation when we started funding that development of that test in 2000 at the University of Pennsylvania. And through our venture philanthropy model, the technology and intellectual property that was funded by the ADDF eventually led to a spin-out in 2005 of a company called Avid Radiopharmaceuticals with about $7 million in venture capital. That company was acquired by Eli Lilly in 2010 for $800 million and became the first diagnostic test approved by the FDA in 2012, beating General Electric and other large companies to market. The reason that Lilly bought the test was they wanted to know if the people that were being enrolled in their clinical trials for an anti-amyloid vaccine actually had amyloid in their brain, because they were having patients enrolled without actually having any biomarker test. And it turned out that 35% of the people that were being enrolled in these clinical trials had negative brain scans. They didn't have amyloid in their brains, which was the target of the drug, and they didn't have Alzheimer's disease. So of course, how could you have success? Now that changed when Biogen came along. You're probably all familiar with Biogen. It's a large biotech company, and they had another amyloid vaccine, but for the first time they used the test that ADDF helped to develop to make sure that everyone who went into the trial had a positive amyloid PET scan. And they also used the test to see if the drug worked, because you could do serial amyloid PET scans and see if the plaque signals was going away. And what they showed was that their drug worked very effectively in what it was supposed to do. At 18 months, 90% of the people had negative brain scans, and the drug completely removed all the amyloid from the brain. Of course, that made it possible to really ask the question, if you remove amyloid from the brain, does that mean that people will get better or that the rate of decline will slow? And that's something that's turned out to be kind of positive in looking at that we've seen that people in fact look like they are slowing their rate of decline. Mark mentioned our biomarkers program, and I'm proud to say that working with the Gates Ventures people, we're going to have a blood test on the market in about 3 years. And that's going to revolutionize Alzheimer's disease the same way that a blood test for cholesterol revolutionized heart disease in every way, including prevention, by the way. The brain scan that we developed showed, and when scientists started using that in 40-year-olds and 50-year-olds and 60-year-olds, that Alzheimer's disease, while it's clinically expressed as a disease in old age, average age of dementia onset about 75, the disease itself actually starts around age 40s and 50s. People start developing positive brain scans at that age. Beyond amyloid, the Alzheimer's Drug Discovery Foundation has a broad portfolio based on the biology of aging. Aging is the leading risk factor for Alzheimer's, and we're developing anti-inflammatory drugs for Alzheimer's disease because there's inflammation in the brains of patients. We're developing what are called epigenetic drugs. We're repurposing drugs for diabetes, which is a risk factor for Alzheimer's. Insulin resistance increases in the brain. The brain is totally dependent on glucose for energy. So we're repurposing and testing diabetes drugs at the Imperial College of London in collaboration with General Electric, Novo Nordisk, the Medical Research Council of the UK, just as one example of how we partner. So we're looking at, ultimately, the future of Alzheimer's is going to be a blood test, and it's going to be like cancer, where we have multiple drugs. Cancer patients are on an average of 4 or 5 drugs because we have to hit many targets within the cell. And we view the future of Alzheimer's treatment will be 2, 3, 4 drugs that'll hit multiple targets, and we're chipping away at that. And as I said, I've never been more excited about the work we're doing. Now, today, we're going to hear from Dr. Ron Crystal. We've been working with Dr. Crystal over at Weill Cornell for about 5 years, and he has a really important program that he's going to tell you about Dr. Crystal is testing whether he can change through gene therapy the major genetic risk factor, not a mutation that causes the disease, but a major genetic risk factor. In 60% of the people that have Alzheimer's disease, he's going to tell us about a molecule called ApoE4 and how he's going to fix that molecule with gene therapy. Dr. Crystal is the professor and chairman of the Department of Genetic Medicine at Weill Cornell, where he's also the Bruce Webster Professor of Internal Medicine, the director of the Belfer Gene Therapy Facility, and a physician at New York Presbyterian. He's a pioneer in the field of gene therapy. He's started several gene therapy companies over the last 30 years, and he's made many breakthroughs in gene therapy that are really just amazing. So please join me in welcoming Dr. Crystal to the podium. Thank you.
Thank you, Dr. Phillip. So you heard what Dr. Phillip talked about in terms of what Alzheimer saw back at the beginning of the 1900s, were these amyloid plaques in the brain. But why do some people get it and other people don't get it? And that's partly dependent on who your parents were. Because there's a gene called apolipoprotein E, which carries cholesterol. It's made in the liver, it's made in the brain. And there are three forms of ApoE: ApoE2, ApoE3, and ApoE4. Most of us are ApoE3, and those of us ApoE3 have an average risk for Alzheimer's. But if you're unlucky enough to inherit the ApoE4 gene from both parents, a so-called ApoE4 homozygote, you have a 15 to 20-fold increased risk for the development of Alzheimer's. However, if you're lucky enough to inherit the ApoE2 gene from both parents, then you have a decreased risk for Alzheimer's. And what got us interested in this was that the individuals who inherit from one parent ApoE4 and from the other parent ApoE2, the ApoE2 takes away the E4 risk. So then the question was, can we change the brain from an ApoE4, for those who have the bad gene, the ApoE4 gene, can we change the brain to an ApoE2 E4 brain? And the way we're doing this is with gene therapy. So gene therapy is a strategy. It's basically a drug delivery system where we're treating with the gene, and the way we get the gene in is we use a virus. And the viruses that we use, we modify so that we take out all the bad parts of the virus, and we're using the virus like a Trojan horse to carry the gene into the brain. So we're taking individuals who are E4 homozygotes, so they have both the E4 from both parents, and we're administering a virus to the brain, and we do that, I'll explain that in a second, and with the ApoE2 gene to deliver the ApoE2 gene directly to the brain. And we do that simply by administering it through the back of the neck and the cerebral spinal fluid. It's an outpatient procedure. It takes about 1 hour or so. And the strategy is, can we convert the brain from ApoE4 to ApoE2E4, therefore taking away the risk for Alzheimer's? So with the support of the Alzheimer's Drug Discovery Foundation, we were able to take this from mice, where we showed that it worked, to larger animals, where we showed that it worked, to manufacturing the drug, the virus that we can put into humans. And we already started the trial. We've already treated 2 patients. Our 3rd patient is for next week. And it's moving along, and be happy to answer any questions about what we're doing. So thank you.
Yeah. So just thought we'd have a little discussion, and if anybody has any questions, please feel free to interrupt, raise your hand, and we'll get right to you. Okay, we have a question.
Yes, the gene therapy that you described, is that administered on patients that also have the amyloid already apparent in the brain?
So that's a very good question, because what we'd eventually like to do is prevent the development of Alzheimer's. But the FDA, because one of of course, when you're developing a new drug, one of the critical issues is safety. The FDA asked us to start with people that already have early cognitive decline or actually Alzheimer's. So the initial individuals are individuals who are ApoE4 homozygotes who already have early signs or actually Alzheimer's. Eventually, should we show that it works, the obvious thing is what you're suggesting is prevention. Yeah, so-
And I think it's fair to say we don't really know exactly why or how apolipoprotein E is a risk factor, genetic risk factor, because it does so many things in the brain. But one way you might want to comment on is the interaction with amyloid, and the other is through cholesterol and how cholesterol maybe prevents.
Yeah, there are multiple. So we're essentially ignoring what all the mechanisms are. What we're doing is more proximal to that. We're basically dealing with the genetics. We know the genetics. There's extensive studies in the genetics that ApoE4 is bad and ApoE2 is good. There are studies that show that ApoE interacts with inflammatory cells, and that may play a role. And some interact with what are called the tau tangles. Some data that suggests that it interacts with the amyloid. But from the approach that we're taking, it's sort of independent of all those mechanisms. We're dealing with the genetics, and if the genetics are right, we should be able to help individuals with Alzheimer's and also prevent it.
Ron, you've actually developed gene therapy for other diseases that are approved by the FDA and actually used. And I know it must seem scary to people that we're going to inject a virus into all these millions of people with Alzheimer's disease. Could you tell us a little bit about some of the success you've had in bringing gene therapy to market already?
Yeah, so gene therapy is the concept is really quite simple. We want to deliver the gene to cells in the body to genetically modify those cells. And the question is how do you do it? You can't just eat a gene and like a pill, and so we use a virus, and the virus interacts with the cell and delivers the DNA new gene directly to the cell, and therefore it functions it. Okay, and so we've had success in a number of areas. In the CNS area, in rare genetic disease of children, where we've delivered the gene and we can show that we can slow down the progression of the disease. There is now a approval for gene therapy for a rare eye disease where people actually are blind and then now can see. We're injecting gene directly with a virus directly into the retina behind the back part of the eye. There's several gene therapy strategies for cancer now. You've probably heard about CAR T, where you modify immune cells to directly target the cancer cells. And so gene therapy, you're going to hear a good deal about over the next 10 years with multiple approvals of therapies.
And I remember I was so impressed last night at dinner when you said that you developed this treatment for Batten's disease, these poor children that have this very serious neurodegenerative disease, and now in your clinic you're actually giving children the gene therapy that you invented. That must be very gratifying.
Yeah, now we know that we're not concerned about safety because these viruses that have been used in thousands of patients now, and the FDA feels very comfortable about the use of these viruses, which have been well studied. And we're actually essentially just using these as delivery systems to be able to deliver the normal gene to correct the abnormality. In this, and for Alzheimer's, the case would be E4T2.
Ron, you're not only a world-leading scientist in this area of gene therapy, but you're also an entrepreneur, and you've started several companies that have been successful, and we're thinking about, or you're about to start a company for this Alzheimer's gene therapy. Could you tell us something about why, as a professor at Weill Cornell, you kind of had to go in that direction to bring these therapies to people?
Well, the challenge is developing a drug is very, very complex. It's one thing to just show initial studies to show that it works, but you have to do large numbers of studies. You have to be able to convince the Food and Drug Administration to be able to approve the drug for therapy, and then you have to manufacture the drug, and that's not something in the academic world we can do in large amounts. And so the obvious thing to do is have partnerships with biotech and pharmaceutical companies, and that's what we're doing. I've started a company called Lexeo Therapeutics, but the whole idea is to be able to move it now so that we really can treat patients and get it to market so it can be widespread use.
And also for raising money, because as you say, this is so expensive. And we were talking earlier about how hard it is to raise money in industry or in these early stage startup biotechs for Alzheimer's disease as opposed to hair loss, for example, and how the venture capital people find Alzheimer's drug research very risky and don't want to put their money there, which kind of justifies the role of ADDF.
It's also, in the academic world, most of our funding for our research comes from the National Institutes of Health, which is governmental money. And they have very large budgets, but it's mostly of what they fund are basic research. And there's no real good place for developing drugs in terms of raising money. And developing a biotech company is be able to bring in venture money, big pharmaceutical money as well, to be able, because it's very costly to develop drugs. I mean, to develop a drug for Alzheimer's is hundreds of millions of dollars.
Billions.
Eventually. And so the only way really to do that is commercialize it.
And I just want to say, before I go to the audience, that we've made over 140 investments in small biotech companies to develop drugs for Alzheimer's disease over the last 20 years. Many of these companies weren't able to get investors to help them advance their drug, and there was a great need for this kind of venture philanthropy funding. I think you had a question.
I did. I'm interested in clinical trials that
Mhm.
have what constitutes the ability of the person to become a part of a clinical trial. Do they have to be in the beginning stages of Alzheimer's or advanced stages of
Yeah.
Alzheimer's?
So let me repeat the question. I think it's fair to say the question is, how does a person get into a clinical trial, and what stage of the disease are most clinical trials focused on these days? As I mentioned, we're supporting about 25 clinical trials right now, and more than half of them are at a stage called mild cognitive impairment. So the stages of Alzheimer's, which are actually recognized by the FDA through a recent guidance, is the first stage is people are biomarker positive without symptoms. This is almost a prevention model. And these people would have, for example, a positive PET scan but have no symptoms. The second stage are people that have a positive biomarker, either a spinal tap or a brain scan or maybe ultimately a blood test, where they have the very earliest signs of symptoms, something called subjective memory impairment, where they're complaining of symptoms, but they actually can't be demonstrated by a doctor, let's say, that they have impairment. It's maybe the spouse thinks there's something wrong or the patient does, but there's no documentation. The third stage is probably the most important stage, which is mild cognitive impairment. And this is the early stage of symptomatic Alzheimer's disease, where people have memory loss. It's obvious to everyone. They might have impairments in what it's called executive dysfunction. They have loss of insight. They have loss of emotional control. They can't plan. They have problems with abstract reasoning, change in personality. And that mild cognitive impairment stage is very important, though, because those people have very few functional deficits. They can still drive and they can do things. And then the fourth stage that the FDA recognizes is dementia, either mild dementia, moderate dementia, or severe dementia. Now, more than half the trials today are in mild cognitive impairment and in the mild dementia stage. And the reason is that we see mild cognitive impairment as a stage where we can clearly identify people that have the disease on brain scan and are affected clinically. And it's also a prevention type of model, because if people come in with mild cognitive impairment and we can prevent them from progressing to dementia, then that would be a big win. So there are drugs on the market today that are for people with moderate to severe dementia, but there are very few programs in clinical development today for those patients. So most of the time we're looking for the earlier stages of the disease.
And it's very important in terms of the question, in terms of clinical trials. If there's no research, there's no cure. And animal models just do not mimic Alzheimer's disease. And so you have to do the studies, as long as it's safe, you have to do the studies in humans. And so it's very important. It's always been remarkable. I've been doing clinical research for several decades, and it's always remarkable how people contribute by participating and having the family members participate, because it's only through that we're going to be able to develop drugs to cure this disease.
Yeah, that's a great point, Ron. And actually, only less than 5% of people with Alzheimer's disease actually go into clinical trials. And the recruitment of patients into trials represents about 50% of the cost of running a trial, finding the patient, screening 10 people to find one patient who's eligible for the trial, and then keeping that patient in the trial. So if an average phase 3 clinical trial for Alzheimer's costs about $400 million in industry, and the companies need 2, 3, 4 trials where they're risking billions of dollars. Half the cost of those phase 3 trials are in trying to find the patients and get them in. And that's why early diagnosis with a blood test is going to make such a huge difference in this, because it'll make the screening procedures and the enrollment so much easier. Any other questions? Yes?
I just wondered if, first of all, you've raised so many interesting points, loads of questions. But first of all, which is the diabetes drug that you're testing? And secondly, are these new drugs like Repatha, which look at the cholesterol in the brain, are they of any assistance? And thirdly, what is the truth about the theory I've heard that if you've had cancer or there's cancer in your family, you are unlikely to develop Alzheimer's?
Okay, I think we're going to have trouble remembering the 3 questions. Well, I can start with the diabetes drug, if you don't mind. So the name of the drug is liraglutide. It's what's called a GLP-1 agonist. It's a drug that's marketed by Novo Nordisk at present, and it's a drug that improves insulin resistance in the body. That's how it works for diabetes, but also in the brain. So the brain is only about 3 to 5% of the body weight and uses at any given moment 25% of the body's energy. And almost all the energy that the brain uses comes from either glucose or oxygen. And the dependence of the brain on glucose is made very clear. If anybody knows a diabetic, you know that as soon as the blood glucose drops below about 60, the person becomes unconscious immediately. And then as soon as you can have them suck on a lollipop and get a little bit of glucose, they wake up. With aging, we all become somewhat insulin resistant. And so one of the theories about Alzheimer's disease is this theory about insulin resistance in the brain at the blood-brain barrier and so on. And so that's kind of the rationale for why we're using liraglutide for the treatment of Alzheimer's. And it's also known, by the way, that diabetes is a risk factor for Alzheimer's disease. So I hope that answers the question. It's being done at the Imperial College of London, and we'll probably have a readout in the next 6 months. And some of the other drugs that are in a similar class, like Byetta, have already shown positive results in other age-related neurodegenerative diseases like Parkinson's disease. And it's an example, if I may say so, of how we use repurposing, a term called repurposing, to develop drugs. So we'll take drugs that are already on the market, where we know the safety and the efficacy and the mechanism of action, and we use those drugs to see if they'll work in Alzheimer's disease. I can prescribe them as a practicing physician off-label, and we can also try to find marketing, commercialization strategies for getting those drugs to market so that they become more available to patients. I think the other questions were about cholesterol, and I forget the third one. So. Oh, with cancer. Maybe you want to.
Cancer, yeah, you can answer.
Okay.
But in terms of cholesterol, it's so the ApoE gene plays a much more subtle role than just lowering cholesterol. There has been interest in the field in terms of using cholesterol-lowering drugs, have not been very successful. So the idea was everybody take Lipitor and you won't have Alzheimer's. That has not been successful. But one of the other challenges of developing drugs for Alzheimer's is the blood-brain barrier. And that is that the brain protects itself from molecules getting from the blood getting into the brain. So small molecules like glucose get into the brain, but bigger molecules don't because of this barrier. And so that's a real problem for drug delivery. For gene therapy, we're circumventing that by putting the gene directly into the brain, and so we don't have to deal with the brain-blood barrier. But the cholesterol-lowering drugs have not worked in terms of Alzheimer's.
So about 15 years ago, an investigator came to us and said they wanted to test Lipitor for Alzheimer's, and they couldn't get any money for it. And they had gone to Pfizer, which had Lipitor, and they went to Merck that had Zocor, and Merck and Pfizer wouldn't help them. So we gave him $750,000 to start running a small clinical trial of Lipitor for Alzheimer's. And the reason he picked Lipitor was because it actually didn't get in the brain, and because we were concerned that lowering cholesterol too much in the brain might be detrimental, and he wanted to just test it that way. When Pfizer heard that there was going to be a clinical trial of Lipitor, They came in and put about $10 million into the study. We enrolled 3,000 people, and as Ron said, it turned out that in that trial back 15 years ago, Lipitor was not effective as a treatment for people with mild dementia. But I go back to the brain scan. We don't know who the people that were enrolled in that trial really were. And I think if it was being done today, we would re-enroll that patient and maybe run that trial over again. We're also testing some drugs that are actually antiviral drugs that are used for AIDS that actually have an effect because cholesterol is uniquely made in the brain. The blood cholesterol doesn't really get metabolized in the brain. The brain makes its own cholesterol, and there's a biomarker called 24-hydroxycholesterol, which is brain-specific cholesterol that you can read out in the blood. And so we want to see the effect of lowering cholesterol in the brain as a treatment because it can have effects on amyloid. This is all hypothetical, but I'm just saying that there are other ways to get at cholesterol in the brain than just the statins. As far as cancer, that's sort of an epidemiological finding. So epidemiology means that you follow people over time and you make observations, and then you are able to get causation. Now, my best example about the pyramid of evidence is that the highest level of the pyramid of evidence scientifically is a randomized controlled trial, which we're talking about. And then the second level is kind of these epidemiological studies. And an example of that, a good example of that is smoking. There's never been a randomized controlled trial to see if smoking causes cancer. That would be crazy, right? There's never been a randomized controlled trial of whether parachutes work, because there's never been crazy people that would jump out the plane without a parachute to see if, the control really worked. But there's enough epidemiological evidence that smoking causes cancer that we recommend against smoking cancer. But there's been other studies to show that bread is bad for you. And how was that done? Well, it was shown that 95% of all felonies are committed within 24 hours of eating bread. Now that's what's called a cross-sectional study. You look at people in time. But I can tell you another longitudinal study, which we think in epidemiology has more value, and that was actually a study of South Beach in Miami. And the demographers looked at the population back in 1980, and at that time, 95% of the population in South Beach was old Jewish women from New York. Then they looked at every 5 years over time, and by the time it was 2015, 95% of the people in South Beach were young, healthy people from all walks of life, let's say young Hispanics. So the conclusion in the newspaper on USA Today was that if you moved to Miami in the 1980s, you started out as an old Jewish woman, but by 2015, okay. So you get my point about the level of evidence, and it's kind of that level of evidence on the association of cancer with Alzheimer's disease. Okay.
Let's take the microphone to you with the questions.
Okay.
Here, and then I'll bring the mic to you so
Okay.
everyone can hear.
When you talk about APO4 and you say it's genetic, and if your parents had it, they gave it to you, what happens if you are an older person and your parents have left the earth? How do you find that out?
Right. Well, how do you find out whether you're APOE4?
Yes. No, well, how do you find out if your parents gave it to you? You say
Well, you
that.
have no other choice. Right? You can't choose your parent. You can't choose your parents. Right? And so if you're an APOE4 homozygote, if you have two, there is no question you got it from your parents. That's the only way you can get it. And as you know, the genetic code is made up of letters. The difference between APOE3, which is the average, and APOE4 is a single letter. So one single letter in the entire gene gives you this high risk, and if you have a single letter difference, it gives you a decreased risk. But there's no question that you get it from your parents. In terms of getting tested, the test actually is available. Your doctor can order the test. There's companies that provide it. Or you can simply go to 23andMe, the commercial test, and we have many people that have contacted us from around the country who say, well, I'm an APOE4 homozygote, and they learned it from 23andMe.
And if I can add, I was talking to a patient just the other day about your exact question. So the question was, suppose my mother and my grandmother got Alzheimer's or dementia, back in the day, and I'm curious if I have it, and what does that mean? So even before we knew about ApoE4, we knew that from epidemiological studies, that if you have a first-degree relative, a mother, a father, grandmother, grandfather, who had Alzheimer's disease, your risk is 4 or 5 times the average population, and if you have 2 family members that got the disease, then your risk goes up to 10 to 15. But in those days, we didn't have ApoE testing. So if somebody comes into me and says, I check a history, and they tell me that their mother had it and their grandmother had it, what I'll say to them is, do you want to know your risk? We should do your ApoE4 testing and see if you have the ApoE4, because one of the only things really that is the genetic risk factor that we know about is the ApoE4 gene. There's no other genes that we know about that have the degree of risk. There's some genes that have a little bit of risk, but they're really not clinically significant individually. So if you have family members that have had Alzheimer's, your risk is increased. If you want to know if you have ApoE4, as Ron said, you can just take the blood test. It goes to Quest or LabCorp, and they can do it. Yes.
I think I was next.
I'm sorry. Okay.
So you talk about the risk factors, and my understanding is, so prevention,
Right.
okay? I know there's a lot of discussion about diet and what diet and
Right.
exercise can do. So if you could talk a little, I read about this mind diet, so-called, and I'd love to know your thoughts about that.
Yeah, prevention has come an incredibly long way. So we used to say that, a heart-healthy lifestyle and heart-healthy, what's good for your heart, the soundbite, what's good for your heart is good for your brain. But it was based on, again, a lot of epidemiological evidence. So it was associations, associations of people who ate blueberries had a little bit less Alzheimer's disease. But a good friend of mine, Dr. Mia Kivipelto in Finland, had the idea to actually run a randomized clinical trial of prevention for older people to see if it prevented Alzheimer's. And what she did was she ran some people through just a single episode of counseling and told them, exercise every day and this kind of thing. Eat a healthy Mediterranean diet, don't smoke, manage your diabetes, manage your hypertension, and let them go. And they would come back a year later and see where they were. And she did cognitive testing and brain scans to see if the rate of the shrinkage of the brain. And then in another group, she had a very intensive behavioral intervention where she made sure that people were compliant with the prevention recommendations that were being made, this multimodal, multi-domain prevention program. And what she showed in this randomized trial was that prevention really works. And we know that. And now that study has gone worldwide. So there's a Japanese, this was done in Finland, there's a Japanese study, there's a US study, it's gone. So that's one thing that we can tell people with fairly good confidence, that what's good for your heart is good for your brain. If you lead a healthy lifestyle, if you manage your comorbidities, if you don't smoke, that you will have an impact on delaying the onset of Alzheimer's disease. The other thing about prevention is with the brain scan, as I mentioned earlier, we can find people before they're symptomatic and start to treat them with drugs. So there's already been a clinical trial which is ongoing of people that are in their 40s and 50s and early 60s who have, we find them by doing brain scans and finding people that have positive amyloid scans and then giving them an anti-amyloid drug to see if we can actually prevent the development of symptoms. That's treating those stage 1 people by the FDA recommendation that I mentioned to you earlier. And the last thing that we're doing right now is trying to combine what Dr. Kivipelto is doing in Finland with some drug interventions to see if by a combination of lifestyle and comorbidity management and drugs like metformin that are anti-aging drugs, we can slow down the rate of cognitive decline with aging and the onset of Alzheimer's disease. So there's a lot of excitement now and a lot of data in prevention. And I'll add one more thing, if I may. But one of the things I find interesting about prevention is that it's very clear that exercise is good for your brain. And we did a study about almost 20 years ago now where we took people, normal elderly 75-year-olds, brought them into a center, did brain scans on them, and tested their neuropsychometrics, their memory and so on. And one group came in and they stretched and they went home for lunch and that was the end of it. And another group came into the center and they stretched and then they went for a brisk 45-minute walk 3 times a week. And at 6 months, we repeated the brain scans and the neuropsychometrics. The people who didn't take that walk and remained sedentary, they their brains shrunk at what is considered normal aging, which is about 0.4% in volume per year. Our brains shrink, and our processing speed and our memories decline on neuropsych. The older people who took that walk, their brains, particularly the hippocampus, where Alzheimer's disease starts, got bigger. Their memory and their cognitive function improved. Subsequently, we looked at the biology. How does that work? It turned out that when we exercise, we release a very powerful neurotrophic molecule called BDNF. When I presented the data on the exercise to my board, one of the board members said, Howard, that's great, but I'm going to be a couch potato. I am a couch potato. I'm not taking that walk. Can't you discover a drug that mimics the effect of exercise? And I said to him, we're already doing that, because what we're trying to do is create small molecules out of that BDNF that people can take that would in fact mimic the effect of exercise. So what I'm saying is, what does it teach us about developing drugs if antioxidants and blueberries work and exercise works and so on? Sorry, Ron.
I didn't have any blueberries for breakfast, but I exercise.
Okay.
I think another thing in terms of prevention is keeping your brain active. Come to symposiums like do the New York Times crossword puzzle and so on. Keep active in terms of your brain. I think that's another very important preventative.
Yeah, and the way I think that's obvious is if you take a human being and put them like a soldier down in a dark dungeon, as a captive for 6 months or whatever, we know that at the end of those 6 months, that people, healthy young people, their brains shrink. The brain is very plastic. It is very much like a muscle, and we need to keep our brains active. I think that's really important. Yes.
I had an easy question, which is if you test positive for the 4, what then happens?
Well,
and you're asymptomatic.
Do you want to take that? Okay. So, I really find, I always get that question, why bother to diagnose this thing? Well, if you had cancer, I'd say, why bother? And you say, well, you can do something about it. Yeah, I could get you on a drug that, you'd live 3 months longer and your hair would fall out and you'd vomit, but you want a diagnosis. I mean, people want a diagnosis. Now, some people want a diagnosis and some people don't. So as a practicing clinician, my job is to find out what your values are. What is what we call the value of knowing to you? And then comply with your wishes, and then do counseling around the results of that. Because if you have a disease, most people want a diagnosis. So what is the value of knowing your ApoE4 status to you? And I think it's very clear that some people want to know their risk because maybe they're not living that healthy lifestyle. Maybe they need to do certain kinds of estate planning that they wouldn't do as well if they didn't know they were ApoE4. Maybe they want to make sure that all their documents are in place. Maybe they want to go into Ron's clinical trial if they're homozygote for ApoE4 to get to contribute to research. Or maybe they want to donate to ADDF to contribute to the research if they don't actually want to participate. The other thing is, let's say the value of knowing is you maybe your mother had it, and you want to know, do you have that ApoE4 genetic risk factor? And it comes back negative. Well, that's really reassuring. That would say that maybe mom had it, but maybe it wasn't Alzheimer's because back in the day we didn't do the brain scans, but at least the person would know they don't have that genetic risk factor, and that can be very, very reassuring to people. So I think there are a lot of results of getting the ApoE4 test. And actually, there's been some medical research around that, also around the PET scan similarly, and it really relates to individual value of knowing. Okay, last question. Yes, sir.
This is a simple statistics question, but if you have a parent, a mother with APOE4 gene, is it more probable that transfer would be to a daughter than to a son? Or how does what kind of probabilities do you have?
It's independent of gender. So if one parent is ApoE4 E4, both E4s, then all the children will get it. But if the one parent is an ApoE4 and another ApoE, then it'll be random, and 1 out of 4 of the children will get it. But it's independent of gender.
Okay, thank you very much. We look forward to the luncheon.
Thank you.
Thanks, sir.